Each single bacterial cell replicates many times to generate millions of cells that form a single bacterial colony on the plate. The appearance of these colonies is termed colony morphology. Describing colony morphology is the first part of identifying and characterizing a bacterial culture, because individual bacterial species often form colonies of characteristic size and appearance. The four main characteristics we use to describe bacterial colonies are size, shape, colour, and any other distinguishing features, such as whether the colony surface is glossy, slimy, smooth, or dry. Here we have bacterial colonies growing on a blood agar plate. It's called blood agar because it contains horse blood. Blood agar is one of the most routinely used agars as it contains many nutrients suitable for even the most fastidious bacteria. Note that there are two colony types growing on this blood agar plate. The colony morphology of the smaller colony would be described as white, round, and 2 mm in diameter. The second colony type would be described as grey, of an irregular shape and about 3 to 6 mm in diameter. Some bacteria also cause lysis of the red blood cells in the agar. This is described as hemolysis. The best way to check for hemolysis is to hold the plate up to the light, like so. Look for clearing of the opaque red blood cells in the zone surrounding each colony. Note the plate on the left has complete degradation of the red blood cells. This is called beta hemolysis, while the plate on the right has only partial degradation of red blood cells. This is called alpha hemolysis. Note the greenish tinge around these bacterial colonies.
Characterization of bacterial culture in agar slant can be observed by the amount of growth. It could be slight, moderate, or abundant. Next is the color of colony. Then its consistency. It could be butyrous, viscid, membranous, and brittle. Optical characteristics, which could be opaque, translucent, opalescent, iridescent, fluorescent, dull, and glistening. Form. These forms could be filiform, equinolate, beaded, effuse, arborescent, and rhizoid. Odor. Last is its liquefaction. It could be gratiform, napiform, infundibule, succate, and stratiform. These are some examples of liquefaction.
A liquid medium. A liquid medium, we can measure the degree of growth according to these types, like homogeneous turbidity and turbid. The first tube here is the control tube. The second here homogeneous turbidity. homogeneous متجانس تكون الجروث of bacteria here. turbid يكون along the tube. على طول التيوب هو turbid، معناتها البكتيريا homogeneous تكون متجانسة الجروث مالتها. In the third tube follicle تكون thick growth at the top of the tube، على السيرفس of the tube يكون الجروث مال البكتيريا كأنه هي مثل القشرة على السيرفس مال التيوب only. In the last tube, the growth deposits in the bottom of the tube, sediment in the bottom of this tube. It reckons in the bottom of this tube يكون bacterial growth.